so for our next talk, we have Michael Fenwick, who's a visiting scientist from Steve Ealick's group at Cornell University. And he's going to be talking about structural basis of substrate selectivity of Viperin and antiviral SAM enzyme. So, Michael?
Okay, I want to first begin by thanking the SBGrid for this opportunity to present. This work is from Steve Ealick's group at Cornell in collaboration with Henning Lin's lab. And I will be discussing two crystal structures that provide a structural basis for the substrate selectivity of Viperin and antiviral radical SAM enzyme. So radical SAM enzymes use a 4-iron 4-sulfur cluster to generate a radical from S-adenosylmethionine, or SAM. Typically, SAM undergoes reductive homolytic cleavage of its C5'S bond, forming methionine and a 5' deoxyadenylyl radical. That radical then typically abstracts a hydrogen atom from a target substrate, producing a substrate radical, which then undergoes radical-based chemistry in the active site to form either a product radical, which is then either reduced or oxidized to form the product. The structures of radical SAM enzymes have a partial Tim-barrel core with variable N and C terminal domains. The active site is formed by the inside of the partial barrel. 4-iron 4-sulfur cluster is embedded in the base of the barrel. 3 iron atoms of the 4-iron 4-sulfur cluster are ligated by 3 cysteine residues in a loop that contains a conserved CX3-CX2C motif. This leaves open a 4th iron atom for coordination by SAM in a conformation that's favorable for producing the 5' deoxyadenylyl radical. So radical SAM, the radical SAM superfamily defined by this core fold presently has more than 110,000 members. Humans have 8 radical SAM enzymes from this superfamily, including Viperin. So Viperin was first characterized in 2001 by Peter Creswell's lab at Yale. They identified a 362 amino acid protein that viruses that associated with the ER and was interferon inducible, and so they named it Viperin or Viperin. They noted that the sequence contained the CX3-CX2C motif, suggesting that it was a radical SAM enzyme. They later showed that the N-terminal domain contained an amphipathic helix that's responsible for localizing the protein at the ER. A striking feature of Viperin is its ability to inhibit viruses. It can inhibit several viruses from multiple virus groups. A few years ago, in collaboration with Peter Creswell's lab and Jorgo Modis's lab, we determined the first crystal structure of a mouse Viperin fragment lacking the N-terminal amphipathic helix. We showed that the protein was monomeric and contains the partial Tim-barrel core. Interestingly, the N-terminal domain was disordered, and the C-terminal domain contained both internal disorder and disorder in the final 25 residues. S-adenylyl homocysteine, an analog of SAM, and the SAM cleavage products methionine and 5' deoxyadenosine bound in conformations that are consistent with radical SAM chemistry. Structural alignment of Viperin with another radical SAM enzyme, MOA-A, showed that the two proteins have a similar set of basic residues at similar positions in the partial barrel. In MOA-A, these basic residues bind the triphosphate group of GTP, substrate of MOA-A. This suggested that Viperin could be acting on a triphosphate, or nucleoside triphosphate as a substrate. Using genomic and biochemical studies, the labs of Steve Almo and Tyler Grove at Albert Einstein, in collaboration with groups at Penn State, in 2018 published a remarkable study in Nature demonstrating that the substrate of Viperin is cytidine triphosphate, that the 5' deoxyadenylyl radical abstracts the C4' hydrogen to produce the product 3' deoxy 3' 4' didehydro CTP, or DDH-CTP. They also showed that this product, DDH-CTP, inhibits the RNA-dependent RNA polymerases of 4 flaviviruses by acting as an RNA chain terminator. In our own work with mouse Viperin, we showed that CTP can be converted to DDH CTP, and that UTP can be converted into DDH UTP. Interestingly, our kinetic studies performed by Drs. Dan Su and Dr. Min Dong showed that the K-cat values for CTP and UTP are very similar, whereas the Km for UTP is significantly larger than that for CTP. We also found that the activities for cytidine diphosphate and deoxy-CTP were significantly lower than that for CTP, and that ATP and GTP showed no activity. And these results are consistent with the study headed by the ALMO group. So we were interested in understanding the structural basis for the substrate selectivity differences, in particular the differences between CTP and UTP. So we determined high-resolution crystal structures of a mouse Viperin variant bound to a 4-iron 4-sulfur cluster S-adenosyl homocysteine, which is a SAM analog, structural analog, and either CTP or UTP at 1.5 or 1.9 angstroms resolution, respectively. In contrast to the nucleotide-free structure, the CTP-bound structure shows considerable ordering in the nucleobase binding site and in the C-terminal tail, shown in magenta. So the C-terminal tail folds across the open side of the barrel and contains a P-loop containing an arginine and tyrosine that makes specific interactions with the gamma phosphate of CTP. So altogether, Viperin makes 16 salt bridges or hydrogen bonds with the triphosphate group of CTP, makes 4 potential hydrogen bonds with the ribose and 4 potential hydrogen bonds with the cytosine group. If we zoom in the nucleobase binding site, we see that for CTP, shown here in blue, that the N4-amine makes hydrogen bonds with the backbone carbonyl of lysine 299 and the side chain of cysteine 314, and the O2 atom makes a hydrogen bond with the epsilon amino group of lysine 319. This chemical complementarity is associated with a high degree of ordering, which is indicative of tight binding. In contrast, if we substitute the N4-amine with an oxo group, as we have in UTP, we get steric clashes with, or we get chemical clashes with the backbone carbonyl involving cysteine 314, and N3, which is now protonated, makes a bad contact with the side chain of lysine 319. So this is associated with some structural displacement and increased disorder in the nucleobase binding region, indicative of weaker binding. So these differences in binding likely account for a much higher Km value of UTP over CTP, and thus accounts for the substrate selectivity differences for the 2 nucleotides. In contrast, the structure of the nucleotides near the ribose and triphosphate groups and the surrounding residues are virtually identical. In both cases, the C4' hydrogen is well aligned to receive or to be abstracted by a 5' deoxydenosyl radical. So this is consistent with the similar K-cat values that we see for CTP and UTP. And lastly, we did a multiple sequence alignment of 46 Viperin-like enzymes and compared this the sequence variation at 25 sites around CTP. So many animal Viperins showed absolute conservation or only conserved substitutions at 25 sites, at all 25 sites, suggesting that CTP is the substrate, but there are some exceptions. If we go through the other kingdoms down to bacteria, we see increased variation occurring in the ribose binding site and pronounced variation in the nucleobase binding site. In contrast, the triphosphate binding site is highly conserved in all 46 sequences. So the results suggest that these Viperin-like enzymes are potential sources for new chain terminators or other nucleotide analogs that could have potential therapeutic value, either as antivirals or possibly as anti-cancer agents. So with that, I'd like to thank Steve Ealick and Henning Lin for their guidance throughout this project. I'd like to thank Min Dong and Dan Su for carrying out the biochemical studies. I'd like to thank the labs of Steve Almo, Tyler Grove, Peter Cresswell, Yorgo Modis, Ty Begley, and Dennis Dean for helpful discussions and/or materials that were used. in this project. I'd like to thank NECAT and APS for excellent beamline support, and I'd like to thank the funding agencies, and I'd also like to thank all of you for listening, and I'd be happy to take your questions.
Thank you, Michael. That was a great talk. And again, if you have questions, feel free to send them to me or Carol in chat. One question. So you'd mentioned the ordering of the C-terminal tail and the nucleoside site on ligand binding in the new structures. Do you think that is something that's potentially? Do you think that is protein motion or just order disorder?
That's a good question. We don't know the structure of the tail when it's not bound to the nucleotide. And there is some helical character in the tail. So there's probably some unfolding and folding going on here, but I assume that the helix is ordered even when it's not bound to the core domain. Does that answer your question?
I think so, yeah. And it's always a tricky question to investigate structurally, and it's.
We don't know what's happening with the tail when it's not bound to the rest of the protein.
Yeah.
We only see it when in the static state.
Thank you, Michael. That was an excellent talk.